This is the final part of Chapter 6, the DNA, the Molecule of Life. This is Section 6D. Now, this last section is talking more about biotechnology, which are practical applications to DNA technology. And our opening question is, what is a GMO? And have you ever eaten a genetically modified organism? If yes, describe what you've eaten. So think about that. Has anyone ever eaten a GMO? Well, I think if you said no, I think you'd be surprised because actually a lot of our food sources are GMOs, and we'll talk about that in a minute. So GMO stands for genetically modified organisms, and they are ones that have acquired one or more genes by artificial means. If the transferred gene is from another species, then the organism is called a transgenic organism. So here's a picture of a transgenic goat that carries a gene for a human enzyme called lysozyme. Now, the use of GM organisms in agriculture has been widespread. The majority of corn, soybean, and cotton grown in the United States is genetically modified. So, if you eat corn chips, you probably are eating genetically modified organisms. Now, in 2015, the FDA approved the first transgenic fish for consumption. But for the most part, most of the GMOs we eat are plants. So genetically modified food crops can be produced by inserting a desired gene into a small circular piece of DNA normally found in bacteria that is called a plasmid. So the plasmid then carries a new gene, which then becomes inserted, for example, in the plant's genome. And then the plant expresses that trait from the newly inserted gene. So here's an example of the plasmid, which is the DNA carrier, and here's the gene for the desired trait, and you can actually put that gene into this small circular DNA plasmid, and then the recombinant plasmid is injected into plant cells, and the resulting, it's actually then enters the plant cells, and the resulting is a genetically modified plant. Now, there are some pharmaceutical companies that have produced genetically modified animals that secrete drugs. So once again, you start with a plasmid, you have a gene of interest, you insert it into the plasmid, and then it's a lot more work to actually insert it into an animal, but ultimately you can insert it, and then, for example, you can make a particular enzyme or some type of drug that that the animal itself is making. Now, 6:16 through 6:18 we are not covering, but we are talking about 6:19, and the question is in chapter 5 we talked about genetic diseases, diseases like cystic fibrosis or sickle cell anemia that in those cases are caused by a single nucleotide change. And from what you've learned about DNA and genetic information, do you think it's possible to fix someone's genes? So several diseases are the result of single gene mutations. So that is this idea of gene therapy, and that's what we're going to talk about next. So gene therapy aims to cure genetic diseases. It is the alteration of a person's genes in order to treat or cure a disease by inserting the correct DNA into the cell. And you can imagine the easiest disorders to fix are those with a single defective gene, like sickle cell anemia. But even though those are easier than, for example, diseases that are controlled by multiple genes, gene therapy is not an easy thing to do. So gene therapy begins with the isolation of the normal gene from a healthy person. Enzymes are used to produce an RNA version of that target DNA gene. The RNA is combined with an infectious but usually harmless retrovirus, and then the virus is used to infect a patient's bone marrow cells, transferring the proper gene to a diseased individual. It sounds like it should be very straightforward, and there have been examples of positive results in gene therapy, but there still is a lot of work to do to make it totally safe. So gene therapy has been used to treat a disease called the severe combined immunodeficiency disease. And some children were cured, but unfortunately others died. They developed cancers caused by the retrovirus that is used as the vehicle to get the good gene into their cells. So at this point, although gene therapy is promising, and in some cases it has been used and been successful, there's still a lot of work to be done to make it consistently safe.